Hi everyone, I want to share with you one of my studies on the prevalence of herpes viruses in the Malaysian population and the development of a multiplex kit for rapid identification. I have divided my presentation into two parts. For part 1, we will talk about the prevalence of herpes viruses in the Malaysian population. Globally, it is estimated that economic burden of herpes virus infection is more than USD 35 billion, and it costed about USD 3.9 billion of economic burden in the Southeast Asian region alone. Knowing the prevalence of herpes virus infection is very important because they have been associated with several types of cancer development in humans, as you can see in this table. More scarily, all these herpes virus can be transmitted through human saliva. Although the prevalence of herpes virus has been reported in many populations, but the prevalence of these viruses in the Malaysian population is unclear. Therefore, we have developed this methodology to determine the prevalence of herpes viruses. In brief, we have randomly collected 725 saliva samples from healthy subjects, and the genomic DNA was extracted from these samples. We then perform PCR using different set of primers that targeting specific gene for each herpes virus, as listed in the table, followed by gel electrophoresis for band identification. Lastly, the prevalence of each virus was calculated. As you can see in the figure below, we have high prevalence of EBV in the population, accounted for 66.3%, followed by 48.5% for HSV8 and 41.9% for HSV1. Surprisingly, we have critically low of HCMV infection in the Malaysian population, with only 0.1%. Since the prevalence of EBV is the highest, we then integrate the data into a meta-analysis with about 3,000 subjects from different populations. In a global view, the prevalence of EBV is about 49.5%. However, when divided into different populations, we can see that the prevalence of this virus was much higher in the Asian population compared to the non-Asian population. One possible explanation of this phenomena might be due to strong culture and habit of sharing food and eating utensils. Besides that, pre-mastication in the Asian culture, as you can see in reports below, also facilitate the transmission of herpes viruses, including EBV to infants. As concluding remarks for part 1, we can see the prevalence of EBV, HSV8, HSV1 is high in the Malaysian population. Understanding of the prevalence of these viruses can help the government bodies in formulating healthcare policies, especially those herpes virus-related diseases. As how this virus could lead to the development of cancer is not exactly clear, therefore future studies should focus on this perspective. Now for part 2, I present about the development of a multiplex kit for rapid herpes viruses identification. The infections of herpes viruses will remain dormant and only become reactivated when the host immune system weakens. Therefore, it is very dangerous to immunocompromised individuals, especially who undergoes chemotherapy, dialysis, transplant, and others. Nevertheless, the reactivations of these viruses typically manifest similar symptoms, such as fever, feeling tired, skin rash, and swelling of lymph nodes. So, a rapid detection and differentiation of these viruses are crucial to ensure effective treatment. Although there are several detection kits available in the market, but they are only able to detect up to 2 herpes virus in a single reaction. Therefore, we think that a multiplex kit that is able to rapidly detect up to 4 herpes virus from human saliva in a single reaction, along with an internal control, is pressingly needed. This slide shows you the overall previous procedures in detecting all four herpes viruses using a real-time PCR method is complicated and time-consuming. And here we show how our innovation overcome these problems with a simple workflow where only required to collect 0.5 ml of saliva and mix with same volume of lysis buffer. Then add 5 microliter of the supernatant after spinning down with 15 microliter of the optimized master mix, and you are ready to go. Our hydrolysis assays designed in this study are very sensitive and are able to detect down to 0.001 nanogram of template, as you can see in the left figure, with very high efficiency and highly specific, as you can see in the table. Here, the left figure actually shows you the presence of EBV in real saliva sample, where you can see a green curve that represents the presence of EBV, while the right figure shows you an example of herpes virus negative, where there is only a curve for the internal control. So what is the impact and application of our multiplex kit? Basically, it can be utilized by global researchers and clinicians for herpes viruses identification. It also can assist in reducing healthcare economic burden and at the same time enhance effective treatment. Besides that, it can increase awareness of the public regarding herpes viruses, and it adds significant value to technology, and it is the first multiplex kit that can detect up to 4 herpes viruses per reaction. In terms of regulation, it could enhance government policies. Our kit developments also align with national policies, such as the Malaysian Madani aspirations, the SDG, especially Goal Number 3, and the strategic plan of the Ministry of Health Malaysia. To conclude part 2, we have developed a multiplex kit that can rapidly detect up to 4 herpes viruses in a single reaction, along with an internal control. It can significantly enhance treatment effectiveness and the quality of life of the patients. We believe our multiplex kit would add substantial value to public health. Before ending my presentation, I would like to thank the Medical Research and Ethics Committee of UMS for approving this study and fund support from UMS newly appointed lecturer scheme. Thank you.